Hello, everyone. My name is Danielle Drabeck, and first of all, thank you so much for having me at the International Toxin Talks. I'm super excited to share my work on this platform and to see everybody else's cool talks as well. So I'm generally interested in coevolutionary relationships and how coevolution proceeds in natural systems. And today I'm going to share my PhD work, the majority of my PhD work that I did examining a potential coevolutionary relationship between opossums and vipers. Now, when I talk about opossums and vipers, this is the first interaction people think about. But in fact, I studied this very peculiar interaction where venomous snakes are prey of certain species of opossum. And the group of opossums that do this weird behavior are mostly found in South America, except our Virginia opossum, which is in North America, is also part of that group. They prey on a variety of venomous snakes, and including snakes from the genus Bothrops, and are immune to their venom. So this behavior is apparent for several closely related species in a tribe called Didelphini. These are the large-bodied opossums, and shown there, the sister taxa in yellow, Metachirus, is known to be sensitive to at least one venom. Now, as we know, venom is a complex cocktail of toxins. So we know how these species are coping with some of the classes of toxins. So they have circulating inhibitors for metalloproteinases and phospholipases, but we don't know really anything about how they're coping with other classes of toxins like C-type lectins. And that's what my work focuses on, is the C-type lectins. Today I'm going to focus on a specific C-type lectin called botrascetin. It comes from a snake called Bothrops jararaca, and it targets a blood protein called von Willebrand factor, or VWF. And it causes severe coagulopathy via induction of platelet binding, and it causes thrombocytopenia. In particular, we know that botrascetin binds to the VWF-A1 domain. It binds quite tightly, and it causes its damage to that binding interaction. So I'm interested in this interaction because my former advisor, Sharon Jansa, and her collaborator, Rob Voss, published a paper in 2011 showing that VWF in Didelphini shows phylogenetic signatures of evolving under positive selection in this group Didelphini, and that the sites that are referred to be evolving under positive selection overlap quite well with botrascetin binding sites. So that's obviously suspect. And additionally, the presence of positive selection indicates persistent diversifying selection, which is a keystone of coevolution. So we know, I expanded this work to do some physiological assays on both the large-bodied opossums, two species of the large-bodied opossums, and one species outside of that group, Monodelphis. And we saw that botrascetin these species were physiologically resistant to botrascetin. In other words, botrascetin did not cause platelet aggregation as it does in species that are sensitive to botrascetin. We also expanded this to an E. coli expressed assay. So we expressed VWF in E. coli from several species of Didelphini and from opossums outside this group, and showed that they have all lost their binding affinity for botrascetin. So here, what you're looking at here is relative binding to humans. So 1 is human binding, and then 74 would be 74 times binding loss, and NDB is no detectable binding. So the take home here is that all of the opossums, both inside Didelphini and outside Didelphini, have lost their affinity for botrascetin. Even Didelphis aurita, we know, has that 8-fold binding loss. We know is physiologically resistant, so we know that that's enough to confer the lack of a physiological interaction. So we have all this cool evidence for coevolution. We have this really cool behavior and a bunch of physiological assays and molecular evidence that suggests that there's coevolution going on. But how can we really know if VWF and botrascetin are co-evolving? This is a canonical problem in coevolution. We have a lot of these matched phenotypes And lots of traits that look like they've evolved together, but this does not necessarily mean coevolution. Many of these traits that have thought to be co-evolving upon further examination are actually not or have been shown to not be co-evolving. So they're either one was pre-adapted or only one is really changing and the other kind of stays static. So without observing ancestral forms, how do we really know that this is happening? We need a time machine, really. And so I'd like to show you some work today that I've done integrating some phylogenetic inference with old and new biochemical and biophysical methods to begin to observe the function of ancestral traits and really get closer at answering whether the system, which appears to have these matched traits, are truly co-evolving. So even if we just look at one side of a coevolutionary interaction, if we look at or are able to observe ancestral function of traits, it can really help us decipher different histories. So for example, if we look at a trait and we see that in its past, there's really no change over time. So for this case, we're looking at rochester and VWF interactions. We see here that there has really been no change over time. These guys were resistant before the vipers even invaded South America, and they kind of stayed resistant. So they just happen to have this VWF that doesn't bind. If we test ancestral phenotypes, we can also reveal other hidden histories. So for the same tip taxa, we might see evidence for an adaptive scenario. The first being arms race coevolution that I will show here, or I'm showing here. So again, same tip taxa, same tip traits, but in the nodes, we're seeing a change that is going gradually, optimally, or towards one optimum trait value over time. And that's indicative of adaptive change towards a single trait value. So we see this in arms race coevolution, and it's typical of large traits like predator prey speed or body size, where the species that are interacting are pushing each other to be more and more and more of a trait. But this is not the only way that coevolution can proceed. It can also proceed by something called trench warfare coevolution. So this is where there's non-unidirectional change, and the way that things are invoking, or they're invoking resistance by invoking a previous form. So they're toggling back and forth between trait states. So they're like, I'm this resistant, I have this sequence, I have this sequence. And this is more typical of things like protein-protein interactions that can abolish binding just by changing one amino acid pretty easily. So as I mentioned before, we have an assay in which we can heterologously express VWF from extant VWF for extant opossums. But what we need now is a way to observe the VWF's function and its change over time. So to do this, we used a maximum likelihood method of sequence reconstruction and using a codon model inferred ancestral DNA. And once we have that inferred ancestral DNA, we can synthesize that, put it into a plasmid vector, the same plasmid vector that we use for extant species, and actually express ancestral protein. So that is what we did. We resurrected ancestral proteins for all of the nodes in didelphiny back to the base of didelphiny. And that's about 15 million years. So we then used purified botricine, and in fact we have two isoforms of botricine A and B. We have these two components now, so we can mix VWF and botricine, test their binding affinity, and essentially observe what these ancestral phenotypes would be. We would ideally have ancestral venom to test this against too, but because of the complexity of venom evolution right now, we have only contemporary venom protein. So we're not really testing whether ancestral VWF will be resistant to ancestral venom, but we're testing is the pattern and the directionality of that change. So before we look at our results, let's just review what our expectations would be based on what we know about this system and what the ancestral binding data might show us. So again, consider a case where venom is evolving and changing via binding to VWF, but VWF has actually not changed at all over time. So this is a pre-adaptation scenario where opossums have just been resistant forever, and that's really not changing. This is what we would expect to see. The nodes are all just resistant. For an arms race coevolution, again, we would expect unidirectional change, so going from not resistant to resistant, sort of convergently, and also towards one trait optimum to the present. And for trench warfare coevolution, we would expect to see this non-unidirectional pattern. So nodes going from more resistant to less resistant and back again relative to contemporary venom. Now, these are not the only scenarios that explain these patterns, but these are the a priori expectations of these patterns, of these phenomena. So our binding data, what does it say? Our binding results for botricine A show a clear pattern of gain and loss of function over time, so a multidirectional change. For botricine B, it's the same pattern. Interestingly, if you look at the deep nodes there, they're exactly opposing one another, which is what you would expect if there was a diversifying coevolutionary interaction going on, which is kind of cool. We see evidence for coevolution, but also we see a great deal of convergence. So we see each of these nodes is independently evolving resistance to botricine along this branch into the tips. So is this coevolution? It's a big question. We have this large associated trait interactions. We have positive selection, which suggests adaptive function. We have botricine-resistant opossums and physiological assays, and we have shown that VWF has lost its binding affinity for botricine. So we have a molecular basis for that physiological reaction. And now with our ancestral binding data, we can reject evolutionary histories which require unidirectional functional change. So that pattern is consistent with trench warfare coevolution. Now, as I mentioned before, other processes such as evolving to many different C-type lectins or switching their prey over time could also produce this pattern, but this is simply consistent with our a priori expectations of coevolution. And what we really need to show, if the next step in showing whether this is co-evolving is looking at reciprocal change in venom and to see if venom is evolving with the same flipping and flopping back and forth relative to contemporary VWF. Importantly, this is a system that is now developed that can test ancestral phenotypes in vitro for co-evolving traits in a natural system. And I just very quickly want to talk about my current work, because this is my PhD work. I'm now a postdoc, and I'm using the same ideas, convergence and coevolution, and I'm using, instead of looking at one interaction, using genomes to find genes which are under strong selection or show evolutionary rate convergence, to try to find out all the genes or discover new genes that are making up these very complex traits. So that includes things like cave dwelling, sleep, and of course, venom resistance. And with that, I will my acknowledgements up and take any questions. Thank you.
All right, hopefully you can still hear me. Now, thank you for doing that. So there are some questions in the chat already. Whoops, let me make sure this doesn't replay again. Not bad. Okay, so with the VWF receptor, do you know of any trade-offs to the normal function of this protein versus the receptors that do not bind botricine?
Yeah, this is a great question. And I inevitably get this question. I get this talk every time. And I've always wanted to have like a project in the back of the freezer looking at this, but the short answer is I don't know. The long answer is I really do want to explore this, because VWF opossums do seem to have some weird coagulation stuff, the way that their their platelets respond, and their normal platelet levels are a bit higher than other mammals. So it's certainly possible. We don't know. At some point in my career, I hope to hash that and figure that out.
There's always so many things.
Yeah.
Steve asked, could VWF theoretically be exploited as a novel therapeutic for snakebite?
Yeah, that's an interesting question. She's gonna answer for me. My knee-jerk reaction is no, but I know that heterologously expressed VWF is used for other things, other therapeutics, because it binds to collagen, and it is biologically active. So I don't know enough about what it would do to other systems to say whether you could really use it for snakebite. And also, I don't know how much and how many species of snake bites there are where C-type lectins are the things that are causing the majority of the symptoms. It's usually metalloproteinases and other things like that. So I think for that one, I'm going to stick to, I'm not sure. I don't know. Yeah.
Any other questions in the chat? Let's ask. Okay. Well, thank you again for doing that. Very much appreciated. Really cool stuff.